All right, in this video, I'm going to be going over scenario 2 for some of these gene expression problems. Titled, Does Food Affect Genes? So the scenario is toddlers such as Malcolm and Matilda can be pretty picky eaters. Malcolm only eats avocados, eggs, cheese, and yogurt, a diet full of proteins and fats. So right away, I'm just going to write for Malcolm, he's eating a diet full of proteins, full of fats, just so I have that down. Matilda only likes to eat noodles, apples, rice, and crackers, a diet full of carbs, but minimal in proteins and fats. So it's almost like they're opposites right here. So then it's saying, all right, using the chromosomes below, develop a model to explain how the genes in the intestinal lining cells of the two toddlers would differ based on the foods they prefer. First thing we need to do, complete the key above. All right? So if a gene's turned off, I'm going to represent that as an X. Right? And if a gene is, and that means that that cell would never express that gene. But if the gene is repressed, that just means it's currently off. Right? I'm going to circle it red. Or, because I understand that not all the time you're going to have like a colored pen or pencil to work with, we could just put an X down. Or sorry, a dashed line. Dashed line. Right. And if the gene is activated, we could circle it green. But if we don't have colored pens with us, we could always just give it a check mark. Okay. So step 1 done. Now, so for each gene, show on the chromosome whether it's permanently off, repressed, or activated. So for each gene, we need to put one of these things down. So we have an intestinal lining cell for both situations. Right. And I'm going to start at the first gene right here. So actin, smooth muscle type. And you do not have to remember all these genes, just use your yellow sheet, right? You should have this, and you'll also have it on the summative as well. So if I'm talking about actin, right, it's involved with muscle fibers, it's only in muscle cells. I'm talking about an intestinal cell, so it looks like for both of these, this gene is turned off. Right. Then we have a cellular respiration enzyme, right? And we can kind of start thinking already, like cell respiration, that happens in like almost every cell except for red blood cells. So if it happens in all cells, and this is an intestinal lining cell, that's a cell. So I'm going to circle both green. Both of these things are going to do cellular respiration to get energy. Now, again, if I don't have that color, I could always just put a check mark here as well. That works. All right. My next gene is lactase. Okay. So lactase, right, is going to be in the intestine cells, and it's required for the digestion of lactose, the sugar, and milk. So it looks like this is going to be a gene that's going to be on in both of these instances. But if we look at Malcolm's diet, he only eats proteins and fats. So it doesn't look like he's going to be getting any of this sugar. Oh, actually, so for here, even though he's eating a diet high in proteins and fats, he's eating plenty of yogurt and cheese, and that's going to be a dairy product, so there's going to be plenty of lactose there for him. So we're going to say that, yes, like Malcolm is making this. All right. In order to digest that yogurt and milk. All right. Now, Matilda, you could kind of go either way on this. All right, I actually would not be too much of a stickler on this one. You could say that this gene is on or repressed. I'm going to say it's on because it's in the intestine cells, right? And she's eating sugars. It doesn't seem like she's eating lactose. So if you put down that in red that it's repressed, I'd be fine with that. So this one's a little bit of a tough one. All right, so let's go to the next one. And we have got the cell growth controller. So if we look here, the cell growth controller prevents cells from dividing. So it doesn't say anything here that the cell's dividing, so I'm going to assume that it's not, and you can do that. And if you have questions on like an assessment, you could always ask me. But if the cell's not dividing, then that means that gene is on. Right? When it's on, it's telling the cell, hey, like we are not going to divide. Right. So I go to the next one, hemoglobin. Hemoglobin is that enzyme or that protein that carries oxygen, and so that's going to be only in my red blood cells, so that's going to be off. And then insulin is going to be that protein that regulates the metabolism of sugars and fats, that's only going to be in my pancreas cells, so that's going to be turned off as well. All right, now finally we're getting to kind of like the main question here for this specific scenario, where we got this fat breakdown enzyme. So the fat breakdown enzyme is only in your intestine cells, so it looks like it could be in both of these, but Malcolm's diet's high in proteins and fats, so absolutely he's making this enzyme. Absolutely he's making this enzyme, right, to digest these. But it looks like Matilda, her diet's high in carbs. So even though she has this gene, it's just not going to be really red right now because she doesn't need this protein. So we're going to say that this is repressed in Matilda. Remember, we could always put a dashed line if we don't have the colored pens or paper or colored pens or pencils. And the check mark there if we need to. All right. So the last thing that we're going to do is the DNA repair protein. All right. That looks like it's in all cells, and it's almost always on because you always need to make sure that your DNA is in good shape. So I'm going to circle those on as well. Right. So that's really it. You're going to be looking at this sheet to see if the genes are on or would be expressed, right, in the cells that you're looking at. And then you're going to look at the specific scenario to say, all right, well, is it actually going to be on or off, right? So even though it can be expressed in this intestine cell, because this person's not eating any fats, we could say that it's off, okay? Now remember, if you said that the lactase gene was also off, you could do that as well because I said that it was on because lactose is a sugar, right, that it says it right here. And she's eating things that are heavy in carbs, which are sugars, but she's not eating lactose, like it doesn't seem like she's eating yogurt or anything dairy. So you could kind of go either way on this one. The important thing is seeing the difference between these two guys right here. So if I were to say, what's the important gene in this scenario? It's the difference between these two right here. So it's that fat breakdown enzyme. Right. And specifically, describe how the gene activity compares in each scenario. So for Malcolm, we could say that it's active or on, right? And for Matilda, we could say that it's repressed. That's just the fancy word for saying that it's not being read or that gene is currently off. Now, let's say Matilda started eating a diet full of fat, right? She just had a giant cheeseburger from McDonald's or something. You bet that this gene would turn from repressed to active. It would be green. And then finally, just describe how this impacts the gene that gets expressed in this scenario. So really, you're just showing why it's important that this gene is on or that Matilda's off. I'm going to focus on Malcolm. I'm going to say because Malcolm eats a diet heavy in fat plus protein, his intestine cells need to make a protein that helps digest this food. All right. So hopefully going over this example and the thinking behind this example was helpful.